Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Unit 1 for October 2023. Let's begin with the first question. Question 1. Nucleic acids include DNA and RNA. Read through the following passage about DNA. Complete the passage by writing the most appropriate word or words on the dotted lines. A strand of DNA is composed of mononucleotides linked by, these should be phosphodiester bonds, from during, of course, condensation reactions. And during this type of reaction, a molecule of water is formed. Since this is a condensation reaction, water is going to be lost. One DNA molecule has two strands of DNA joined together by, these are hydrogen bonds between complementary base pairs. And the DNA molecule then twists to form a double helix. In part B, the table shows some components of DNA, mononucleotides, and RNA mononucleotides, and shapes that can be used to represent them. We have the bond, we have thymine, there is uracil, of course this is only found in RNA. We have deoxyribose, we have ribose, and then the phosphate. So they said draw one DNA mononucleotide using shapes selected from those shown in the table. Because this is going to be a DNA molecule, I have the phosphate here, we have the deoxyribose, which is going to be D, not ribose, because this is DNA. I used thymine as the nitrogen base because uracil is found in RNA. I couldn't use it among those given here. So this is a representative mononucleotide of DNA. This brings us to the end of question 1. Let's continue to question 2. Question 2. Polyphenol oxidase is an enzyme found in avocados. This enzyme causes green avocados to go brown when they are cut open. The equation shows the reaction catalyzed by this enzyme. So we see there is monophenol, which is a reactant, being converted into O-diphenol, which is the product. In part A, the graph shows the energy changes during this reaction when the enzyme is not present. Remember, when the enzyme is not present, we're going to have greater activation energy. So they say which arrow shows the activation energy for this reaction. Activation energy is measured from the position of the reactants to the highest peak on the graph. So Q is going to be our activation energy. And the answer is going to be a B. Next, they say which row of the table shows the molecule present at time Y and time Z on this graph. At time Y, we have the reactant, and at time Z, we have the product. So if we go back here, this is the reactant, and that is the product. So at time Y, we have the reactant, which is the monophenol, and at time Z, we have the product, which is O-diphenol. So C should be my answer. Here they say which row of the table shows how the length of the arrows P and Q on this graph would change when polyphenol oxidase is present. If the enzyme is present, this is going to decrease, but this will remain unchanged. So Q decreases, P remains unchanged because P is going to be the enthalpy change of the reaction, while Q is the activation energy. So the activation energy is going to decrease, while this one will have no effect. In part B, the graph shows the changes in monophenol concentration in the presence of polyphenol oxidase at temperature 25 degrees Celsius. On the vertical axis, we see the reactant concentration, and on the horizontal axis, we see time. We see as time increases, the concentration of the reactant decreases. So the first part here says calculate the rate of reaction at 3 minutes. They want you to draw a tangent to help you with your calculation. I drew my tangent on the curve at 3 minutes, and I use 0.416 and 1.540 to calculate the rate, which is going to be the gradient of the tangent. So 40 minus 16, which is the change in the vertical components, divided by the change in the horizontal components, which is 9.6 mol per decimeter cube per minute as my rate. Then here they say this reaction was carried out below the optimum temperature for the enzyme. Explain what would happen to the shape of this curve if the temperature was increased to the optimum temperature of the enzyme. If the temperature was increased, the initial rate is going to be faster, so the curve is going to bend in more, or it's going to have a steeper gradient. And again, it's going to level off earlier. So I say the initial part of the curve would be steeper because there would be more kinetic energy for faster collisions between the enzyme and the substrate. And the curve would level off sooner because reactants will be finished faster. So this brings us to the end of question 2. Let's continue to question 3. In question 3, amino acids in the diet of animals are used to synthesize proteins. The rate of protein synthesis is dependent on a number of factors. The graph shows the effect of the mass of protein in the diet on the rate of protein synthesis. On the vertical axis, we have the rate of protein synthesis, and on the horizontal axis, we have protein in diet per gram of protein per kilogram of body mass per day. So we can see, of course, the values are increasing, but after this point, they level off to about a rate of 150. So they say calculate the minimum mass of protein a person with a mass of 70 kilograms should eat in 1 week if they want a rate of protein synthesis of 150 milligrams per protein per kilogram body mass per unit time. Because here they want the minimum, a rate of 150 is reached at a minimum of 1.4 because the minimum amount of daily protein in gram per kilogram based on the graph is 1.4 grams per kilogram. Since this person is going to be 70 kilograms, so the minimum amount of daily protein in grams per 70 kilograms is going to be 1.4 times 70. Since 1 kilogram will be 1.4, so 70 kilograms is going to be 1.4 times 70, giving us 98 grams. But this is going to be per day. You can see it's daily protein. So to get the amount per week, we have to multiply by 7, since a week is 7 days. And I get 686 grams per week, which is going to be my answer here. And in part B, name the 2 stages in protein synthesis in the order that they occur. The first stage is transcription and the second stage is translation. Next, they say describe the role of messenger RNA and transfer RNA in protein synthesis. Messenger RNA is made from DNA, and this contains the information for the assembly of the amino acids in order to create the protein. So it has to leave the nucleus, go to the ribosomes where it's going to be read, and then the transfer RNA will deliver the amino acids in order to produce the protein. So the messenger RNA will have a codon while the transfer RNA will have the anticodon, and so on. So I said messenger RNA contains a copy of DNA which contains genetic information about a protein to be made. Messenger RNA moves out of the nucleus to the ribosomes for translation to occur. Transfer RNA transports specific amino acids towards the ribosome. And transfer RNA has an anticodon which is complementary to the codon on messenger RNA. The codon and anticodon bind to each other as an amino acid is added to the growing polypeptide chain. So this brings us to the end of question 3. Let's continue to question 4. In question 4, red-green color blindness is an inherited condition. The table shows some statistics on red-green color blindness in some countries. So we can see the country, the population size in millions, the number of people who are red-green colorblind in millions, as well as the percentages. The first question says complete the table to show the number of people who are red-green colorblind in India and the percentage of people who are red-green colorblind in Malaysia. So in India, because the percentage is 5.2 and the total population is 1380, so 5.2 divided by 100 times 1380, of course in millions, gives us 71.76 millions, and that's the answer here. And for Malaysia, because we know this is the total population and this is the number of people, so the percentage is going to be the number of people divided by the total population times 100. This gives us 3.2%. And again, remember, this is in millions and that is in millions, so we don't have to consider the millions. And in part B, the percentage of males in Malaysia who are red-green colorblind is 6.7%. And the percentage of females who are red-green colorblind is 0.4%. They want you to calculate the ratio of red-green colorblind males to red-green colorblind females in Malaysia. So for the males, we have 6.7 divided by 100 times 32 million, which is going to be 2.144 millions. And for the females, we have 0.4 divided by 100 times 32 millions, giving us 0.128 millions. So the ratio of males to females is going to be 2.144 to 0.128. Again, this is in millions, which is 16.75 to 1, rounding off to 16.8 to 1. And this is my answer here. Next, they say explain why the percentage of red-green colorblind males is greater than the percentage of red-green colorblind females. If the number of males is greater than the number of females, then it means this is going to be carried on the X chromosome. And since females can have 2 X chromosomes, one could have the allele for colorblindness while the other doesn't have. And that means those who have that trait could be in heterozygous state for females, or there is a percentage that is going to be in heterozygous state for females. But once males have the allele, they're always going to have the red-green colorblind condition. So I said colorblindness is sex-linked and carried on the X chromosome. Women have two X chromosomes, so they can have the faulty allele but only be carriers because another chromosome may contain the normal allele. Women only show color blindness when in a homozygous recessive genotype. In men, because they have one X chromosome, once the recessive allele is present, it will be expressed in the phenotype. So this brings us to the end of question 4. Let's continue to question 5. In question 5, many animals have a heart and circulation. Molecules such as glucose move by mass transport to overcome the limitations of diffusion in meeting the requirements of an animal. State what is meant by the term mass transport. In this context, mass transport is the movement of all components of blood through blood vessels. And then they say all the cells in an animal require glucose. Explain the limitations of diffusion in providing cells with glucose. Diffusion occurs efficiently if the concentration gradient is high. So I say the rate of diffusion may be too slow to provide certain cells with the required amounts of glucose because diffusion occurs following the concentration gradient. And if the concentration gradient is low, then diffusion is slow. For example, movement of glucose into the villi from the small intestines. And in part B, the diagram shows a mammalian heart. They say draw arrows on the diagram to show the flow of the oxygenated blood, meaning oxygen-depleted blood into, through, and out of the heart. Oxygen-deficient blood is going to come from here. Actually, I could also draw an arrow from down here. That's also okay. So it's going to flow in here and in there into, this is going to be the right atrium, and then it goes through the tricuspid valve into the right ventricle, and then move through the pulmonary artery, again here as well as that connection on the other side. And in part C, they say that tunica media is the middle layer of the wall of an artery. The table shows the percentage of some components in the tunica media of arteries of different sizes. So the components include smooth muscles. In the large arteries, we have less smooth muscles, and we have greater smooth muscles in smaller arteries. We have collagen is going to be more in the large arteries. Collagen has that extra strength it gives to the blood vessels, so we expect it to be in the larger arteries that carry blood at very high pressure. And then elastin is going to be present in larger arteries at a higher concentration in comparison to all the other arteries. And this is because as the pressure increases, the blood vessels are going to expand a little bit. So it means they need to have that recoil to return into their original size when the pressure decreases. So elastin is supposed to be in higher concentration. So they say explain why the percentage of the components in these arteries are different. So I said blood flows through large arteries at higher pressure than medium-sized arteries, and it flows through small arteries with the least pressure. So large arteries have higher percentage of collagen to increase strength and withstand pressure. Large arteries have higher percentage of elastin to enable them to recoil. And the smaller arteries have a higher percentage of smooth muscle to control blood flow, reduce friction, and maintain blood pressure. The graphs show differences in some features of blood vessels. So we can see the different features. Of course, these are larger vessels. This is decreasing. This is going to be constant. Increase become constant. This decreases, flattens, and then increases. That is going to have an increase. This is stable or constant, and decreases, and then increases and stabilizes. And that increases, stabilizes, and decreases. So they say which graph shows the diameter of the blood vessels. The diameter of blood vessels is going to be small in the arteries, but it becomes smaller as we approach the capillaries. And then after it widens as it goes through the veins. So this is going to be perfect. It decreases, becomes constant, and then increases. So graph 3 is going to be the answer. And they say which graph shows the permeability of the blood vessels. I would expect graph 2 to be lower permeability, higher permeability in capillaries, and then lower permeability in the veins. So graph 2 is going to be the suitable answer for that. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Starch from plants is used in the production of food, paper, and building materials. The structure and chemical properties of starch depend on its amylose and amylopectin content. The table shows some of the bonds found in amylose and amylopectin. Put one cross in the appropriate box in each row to show whether the bond is present in these molecules. Glycosidic bonds are going to be present in both amylose and amylopectin. The 1,6 glycosidic bonds are going to be present only in amylopectin, and then hydrogen bonds are going to be present in both amylose, and amylopectin as they fold around. In part B, when sources of starch are heated in water, amylose moves into the water. The graph shows the percentage of amylose that is lost from 4 sources of starch in 3 temperatures. On the vertical axis, we have the percentage of amylose lost from the source of starch, and horizontally, we have the source of starch. We can see the key shows us the temperature, and overall, we can see at 80 degrees, there is greater loss in comparison to the others, and at 60 degrees, there is the least lost. So the higher the temperature, the greater the amylose lost. So they say describe 2 conclusions that can be made about the effects of temperature and source of starch on the loss of amylose. Of course, the higher the temperature, the greater the amylose lost from each source. Remember, we are just describing. And then the effect on the source of starch. If we go back here, we can see cassava had the greatest effect and potatoes had the least effect. So here I said cassava lost the most amylose and potatoes lost the least amylose. In part C, the loss of amylose and amylopectin during the cooking of rice affects the hardness and stickiness of the cooked rice. The effect of temperature on the loss of amylose and amylopectin was investigated. The diagram shows part of the method used. So rice was mixed with water and soaked for 30 minutes. The mixture was divided into 9 equal samples, and each sample was heated to a different temperature and the liquid poured off when the temperature was reached. The liquid collected at each temperature was analyzed. So the graph shows the results of this investigation. On the vertical axis, we have concentration of amylose and amylopectin in the liquid, and here we have the temperature. The higher the temperature, the higher the amylose and amylopectin leaking out until 85 degrees. After 85 degrees, increase in temperature causes a reduction in the amount of amylose and amylopectin leaking out. The other observation we see is there is leakage of amylopectin earlier. At 20 degrees, we can see amylopectin is leaking out, but there is 0 amylose leaking out. We also see at 47 degrees Celsius, they have the same amount leaking out. Also at 64 degrees Celsius, we have the same amount of amylose and amylopectin leaking out. They say compare and contrast the effect of temperature on the loss of amylose and amylopectin from rice. So here the similarities I wrote: increase in temperature until 85 degrees led to more loss of amylose and amylopectin. However, after 85 degrees Celsius, there is a decrease in the amount of amylose and amylopectin lost. At 47 degrees Celsius and 64 degrees Celsius, the same amount of amylose and amylopectin is lost. For the differences, below 47 degrees Celsius and above 64 degrees Celsius, there is more amylopectin lost, while between 47 and 64, there is more amylose lost. Also, amylose loss begins after 40 degrees Celsius, while amylopectin loss begins at 20 degrees. We can see this is after 40, and then we see this is about 20 degrees. And next they say the investigation continued by adding a replacement liquid to finish cooking the rice. The hardness and stickiness of the cooked rice was determined for each replacement liquid. The table shows the results of this investigation. So we see water containing amylose, water containing amylopectin, and water containing both. The hardness is going to be high in water containing amylose, as well as high in water containing both amylose and amylopectin. So we can conclude that amylose is responsible for hardness. We also see stickness is going to be higher in water containing amylopectin as well as in water containing both amylose and amylopectin. So since we see greater stickiness when amylopectin is present, we can conclude it's responsible for the stickiness. And then of course water has the least for both. So here they said comment on the effect of the replacement liquids on the hardness and stickiness of cooked rice. They want you to use the information in the table to support your answer. Since I've already explained from the table, I said water had the least hardness and stickiness. Water with amylose caused more hardening. Water with amylopectin caused more stickiness. Water with both amylose and amylopectin led to greater hardness and stickiness. However, the results don't show mean values and no error bars or standard deviation, so we can't comment on the validity of the data. And here they say suggest why rice should be cooked in a measured volume of water with the lid on until all the cooking liquid has been absorbed. Use the information in the table to support your answer. If we cook while placing the lid on, the liquid is going to be maintained inside the cooking pot, and all the amylose and amylopectin is going to be reabsorbed or is going to be retained within the pot. So this is going to give the rice the correct hardening as well as thickness. So I said keeping the lid on maintains the cooking liquid in the cooking pan. All the cooking liquid will be absorbed by the rice, preventing loss of amylose and amylopectin, so the right amount of hardness and thickness is achieved. So this brings us to the end of question 6. Let's continue to question 7. In question 7, a number of factors increase the risk of cardiovascular disease, including blood cholesterol. In one investigation, scientists measured the blood cholesterol level in a group of people and monitored the development of heart disease in the following 10 years. The graph shows the results of this investigation. On the vertical axis, we see percentage of groups, and then here we can see blood cholesterol. From the key, did not develop heart disease, we can see those, and then did develop heart disease. As the concentration of cholesterol increases, the percentage of both groups increases. However, it reaches a peak, and then we see the percentage decreasing. We see there is greater decrease in those that did not develop heart disease. So they say some people interpreted the data as showing evidence for a relationship between blood cholesterol and levels of heart disease. Other people interpreted the data as not showing evidence for a relationship between blood cholesterol level and heart disease. They want you to describe the evidence for and against a relationship between blood cholesterol level and heart disease. Evidence for, of course, as blood cholesterol level increased, until a certain level, the percentage of people developing heart disease increased. But the evidence against, since people with low levels of cholesterol also developed heart disease, some other factor could be responsible. This is true. We can see both increase and then they both decrease. It doesn't make sense that only blood cholesterol is causing the changes we see. In part B, they say other investigations have shown that the level of high-density lipoproteins, HDL, and low-density lipoproteins, LDLs, are significant in determining the risk of CVD. The graph shows the relationship between the levels of HDLs and LDLs on the incidence of CVD. We can see incidence of CVD on the vertical axis and then level of HDL as well as the level of LDL. As we go this side, there is increase in the levels of LDL, and as we go like this, there is increase in the levels of HDL. Since here we have the greatest HDL, and closer here we'll have the lowest LDL, I'm talking about this value here, greatest HDL, lowest LDL, we're going to have the greatest HDL to LDL level. And over here, we will have the least HDL, and we'll have the greatest LDL. It means we're going to have the smallest HDL to LDL level. So down here they say the following conclusions can be made from this graph. As the level of LDL increases, so does the incidence of CVD. We can see here they're talking about LDL as it increases. That's true. As the level of HDL increases, the incidence of CVD decreases. That's true again. And then the higher the ratio of HDL to LDL, the lower the incidence of CVD. This is going to be greatest here, and we can see it's going to be lower incidence of CVD. They say describe the evidence shown in this graph that support these conclusions. As the level of LDL increases, the height of the bar graphs increases, showing increase in incidence of CVD. We can see LDL increasing, or the graphs are becoming higher or taller. And then as the levels of HDL increase, the bars decrease in height. We can see going from the other side to this side, they decrease in height, showing a decrease in incidence of CVD. And then as the ratio of HDL to LDL increase, the height of the bars decrease. The bar height is the smallest at the highest HDL, LDL level, which is this one here, and it's going to be the highest at the least HDL, LDL ratio, which we see as that here. So the conclusions are all valid. And in part C, they say the low-density lipoprotein receptor, LDLR, is a protein on the surface of liver cells that is involved in the endocytosis of LDL. The diagram shows the LDLR in a cell membrane. We can see it there, and this is a cell membrane. Explain how the LDLR is involved in endocytosis of LDLs. This is because the LDLs are going to bind onto the receptor, and then they're going to be held in position. A vesicle is going to be formed around the LDL. Endocytosis occurs, and it's going to be delivered on the other side of the membrane. So I said LDL binds to LDLR and holds it in position. The cell membrane forms a vesicle around the complex formed. The phospholipid shifts to allow the vesicle to enter the cell by endocytosis. The next I say the lower pH inside the cell causes the LDL to separate from its receptor. Suggest how a change in pH could cause this separation. When the pH changes, R groups are going to be charged, so this is going to change the tertiary structure of the protein, and the receptor will no longer hold the LDL in position, meaning the LDL is going to be released. So I said the H plus ions due to low pH causes the R groups to become charged. This changes the protein's tertiary structure, and the bond between the LDLR and LDL breaks, releasing the LDL. And then they say mutations in the gene coding for LDLR can result in an increased risk of CVD. Explain why mutations in this gene can increase the risk of CVD. If there is a mutation in this gene, it means the receptor formed is not going to be functional, and therefore it cannot bind to LDL, meaning the LDL cannot be taken up by the cell by endocytosis. So I said a mutation leads to a change in the structure of LDLR. The receptor can't bind to the LDL because they are no longer complementary. So LDL will not be taken into the cell by endocytosis, and this will cause LDL to remain in blood. If damages occur to blood vessels, then there is a higher chance of formation of atheroma leading to CVD. This will occur because there will be excess LDL in the blood. So this brings us to the end of question 7. Let's continue to question 8. Question 8. In 2021, it was reported that cardiovascular disease, CVD, killed 17.9 million people per year. This is 31% of all global deaths. They say calculate how many global deaths there are in 1 year and express your answer in standard form. So if the total deaths are X, 31% of X is going to be 17.9 million. It means X is going to equal to 5.77 times 10 power 7, and that was my answer. Then next they say, it is predicted that by 2030, more than 22.2 million people will die each year from CVD. Suggest how this prediction was made. When people are predicting the future, they look at previous trends that have occurred, and then they could extrapolate data in order to predict what could happen in the future. So I said by collecting available data of deaths from CVD from previous years, using computer software and considering trends, for example, how people smoking, lifestyle and non-lifestyle factors. So they consider trends in disease progression. The data is plotted and extrapolation occurs or carried out to predict the data for 2030. In part B, the rhizomes of a ginger plant are used in cooking. The photograph shows the rhizomes of a ginger plant. These are the rhizomes. They say these rhizomes have high levels of antioxidants. Some people believe that eating these rhizomes reduces the risk of CVD. Explain why eating these rhizomes may help to prevent CVD. If people eat the rhizomes containing the antioxidants, oxidative stress is going to be reduced, and then the chances of plaque formation in the blood vessels is also going to be reduced, and that reduces the risk of CVD. Then they say there is very little data available to show that these rhizomes reduce the risk of CVD. Suggest why these rhizomes can be eaten to prevent CVD, even though their effectiveness has not been thoroughly tested by scientists. This is because the rhizomes are already food, already consumed by people, so they do not cause any harm. That's why people can still eat them, even without being tested. Explain how a study should be designed to collect valid and repeatable data to show the effectiveness of using ginger in preventing CVD. Here they should use large groups of people. These people should not have any CVD-related conditions, and so on. So I say to create valid results, remember we are looking at valid results, large numbers of people should be used, and the studies should be repeated in multiple groups of people. The repeat experiments should occur among categories of people. These should be similar in all studies. For effectiveness, the group in each study should be the same gender, same age, and should have similar risk factors of CVD. It is better if they are not smokers or they have the same lifestyles. The participants should be given the same daily amount of ginger. Of course, this ginger has to be, if you're using liquid, you squeeze and get out the liquid. It should be the same concentration and same volume. So the ginger should be from the same source or same species. The participants should be exposed to the same amount of free radicals, and then the participants should be monitored for a long period of time since CVD takes long to develop. They should include control groups, and these should be given a known antioxidant, and the results should be analyzed using suitable statistical tests. So this brings us to the end of question 8 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.